Hi, everyone. This is Jeff Gruen, and I'd like to present to you some of the work we're doing on the genetics of learning disabilities, and with particular emphasis on rotation projects for graduate students beginning this fall, 2021, and extending all the way to the spring 2022. One of the major themes in our lab is really dyslexia or reading disability. And this is really quite common. Worldwide, about 10% of students struggle with dyslexia. These are students in regular classes, at regular schools, and with normal or above normal IQs. Between 60 to 80% of reading performance, including dyslexia, is due to multiple genetic variants, so it's a highly heritable trait. But for some children, actually a durable intervention can make a huge difference in their lives and their performance. In these data from Maureen Lovett, published back in 2019 at the University of Toronto, Maureen has identified kids who struggle with reading grades 1, 2, and 3 when offered a very intense, prolonged intervention over 9 months, and then retested 2 years after completion of the intervention to see how durable the intervention has been. And what you can see is when identified early and offered an intense intervention, these kids, about 75% of these children will be reading at grade level even 2 years later. So what Maureen has taught us is that early identification and intervention is really important. And in fact, the emphasis of our lab over the last 20 years has been to develop a genetic screen to enable early identification. We've done this through several important studies, the first one being the GRAD study, or the Genes, Reading, and Dyslexia study, which was conducted at multiple sites across North America and enrolled exclusively African American and Hispanic American children in the only genetic study of dyslexia ever published. And then we followed this up more recently with the New Haven Lexinome Project, a longitudinal study here in New Haven of children in 2 waves in grade 1, who we followed from grade 1 all the way through grade 5, looking at for the genetic component of response to intervention. I'd also like to direct your attention to a poster that we're presenting from Xiying Wang. We're presenting her data on a GWAS, a genome-wide association study, and gene-based analysis of inattention hyperactivity in the New Haven Lexinome Project, here available also on the web, and a poster from Haley De Silva, who is describing Neurostack, a cloud formation tool for analyzing neuroimaging data using AWS cloud computing. So clearly, early identification is important, but what about all these other children and even adults who have problems with dyslexia? And as you can see here is if these children are identified in middle school or high school, at best, maybe 30% of these kids will respond to intervention. In other words, it's not durable at all. Should we ignore these people? Should we ignore these children, who literally are tens of millions of children and adults? Or is there precedent in other models that we can pay attention to, or should at least look at for learning disabilities as a potential other way to address their problems with individuals who struggle with reading? In the United States, the most prescribed drugs for children ages 12 to 19 years are actually central nervous system stimulants used for attention disorders. Overall, 6% of children in this age group are prescribed drugs like Ritalin, Vyvanse, Adderall, and even in the younger children, ages 1 to 11, over 3% are prescribed these drugs. Yet, there are no drugs that have been shown to help with dyslexia. Why is that? Well, certainly not for lack of trying. We've identified many genetic variants over the years and genes associated with both reading and language impairment, and have published these multiple times in multiple venues. But these are genes, and they don't necessarily identify pathways or the metabolic pathways, and therefore we don't know the gene targets, or the potential drug targets. So therefore, no drugs have ever been developed specifically for dyslexia. Now, we've actually been on a path to remedy this through work in human embryonic stem cells to identify these potential drug targets. And I would direct your attention to a poster from Stephen Paniagua, another graduate student in the lab, who presents on the function of KIA-0319, a dyslexia gene in human neuronal development. And what Stephen has done is he's looked at human embryonic stem cells as they differentiate into mature neurons, and the effect of knocking down a well-known, well-published, well-validated dyslexia gene called KIAA0319. And to summarize Stephen's work, what he's shown is that as embryonic stem cells transition into neuroepithelial cells, normally they would express SOX10 at a very high level. But when he knocks down KIAA0319, it actually decreases SOX10 expression. And really interesting, during the transition from neuroepithelial cells to neural progenitor cells, what Stephen has shown is that the normal expression of PAX6 is actually shifted earlier, so that there's an intense but early expression of PAX6. In other words, the hold on this transition into neuroepithelial is released earlier, so that these cells transition sooner into neural progenitor cells than what would usually happen without the knockdown. This is a fundamental shift, a shift that occurs during early human brain development, and it affects both radial, or could affect both radial and lateral migration of neurons during brain development, which would significantly change connectivity in the brain, and which is an underlying pathophysiology for dyslexia and other learning disabilities. So as we tried to illustrate is that in our lab, we use many multidisciplinary approaches, and we go from translational studies, actually clinical studies in children. We do a lot of sequencing. In the New Haven Lexinome project, we've sequenced over 500 children by whole genome sequencing at 30x level. We translate this into what's happening in functional magnetic resonance imaging, specifically to look at connectivity, and we recapitulate our findings, what we're seeing in human embryonic stem cells, with the idea of bringing it back to the clinical setting. Today, I've focused mostly on what's going on in the lab with human embryonic stem cells, and which we'd like to extend into brain organoids as well. And so potential rotation projects for graduate students would be to continue modeling our learning disability genes in human neurons using knock-in, knockout, knockdown of learning disability genes in human embryonic stem cells, as well as brain and cerebellar organoids. To analyze these data, we are doing RNA sequencing. We are planning on doing electrophysiological studies to look at conduction, and also interorganoid connectivity models between organoids. We continue with our other studies in the lab, which are computational. These are in extant, large, well-characterized data sets, including the Adolescent Brain Cognition Development Study, the ABCD study, which is a study of 10,000 children ages 9 through 15, sponsored by the NIH, the UK Biobank of over 200,000 adults, and GenLang, which is an international consortium of children from data sets from Europe, America, and both Americas, as well as Australia. We continue to look at genetic sharing and comorbid disorders as well. So with that, I'll stop and thank you for your attention, and happy to answer any questions you may have. Please contact me through email, jeffrey.gruen@yale.edu, or through our website at yale.edu. Thank you so much.